Thank you. Thank you, Alex, for the introductions, and good afternoon to all from Cambodia. Let me just share my screen here. I hope you see the PowerPoint, not the presenter view.
Perfect. We can see it perfectly.
Okay. Yep. First, I would like to thank for the invitation to share our work here in Cambodia. And I have the pleasure to present today the effort in the zoonotic virus surveillance here conducted in Cambodia by the virology unit at Institut Pasteur in Cambodia. And I take the example of the detections of coronaviruses here in Cambodia. Just before starting, I like very much this slide showing how the spillover events occur, and just to remind us also that we live in a world where there is interface between that people living closely with animals, including domestics and also wildlife. And we should remember also different animals carry their own different viruses and pathogens. And these pathogens might at some point spill over to different hosts and at the end to the humans, causing outbreaks and pandemics that we experience today. And it's also that the way we live, the way we behave, that put ourselves at risk to those potential zoonotic pathogens. And just a few examples here that to illustrate those contacts and interface between animal and humans. And I take this opportunity as well to present a very good example of One Health surveillance that was implemented more than 10 years ago, called PREDICT programs funded by USAID, led by UC Davis. And it's something that we should understand and give value to this kind of program that's long-term prospective surveillance using One Health approach. And unfortunately, this program was ended shortly before the pandemic starts. And this program is very important, very much important for the prevention and the preparedness for any future pandemics. And what is important in this program is that it's a large program that is implemented in several countries in the objective of strengthening lab capacity, also the lab network, and also the way we work together in One Health and to prepare ourselves for any emergence. So in general, this program, this 10 years program focused on a coordinated One Health surveillance strategy by looking at community at high risk where there is a presence of interface between wildlife, livestock, and humans living in the same communities. And additional to that, there is also a syndromic surveillance trying to look at patients admitted to hospital and that might present a spillover or infections that caused by virus found in those animals. And on the top of that, what is also important is to look at the risk behavior, the way people live, the way people think, and the way people practice that might pose them into that help the transmission from viruses from animals, and which is also important also for to find a way to prevent and measure to prevent any spillover from animal to human. Just a few examples of the high-risk interfaces in Cambodia that we have studied during the PREDICT programs. One is the guano farm, where people raise bats to collect guano and to sell as a fertilizer. It's a means of to have incomes for the house, for each household. But the way people work, the way people raise this bat is that they put the bat roots, the palm leaf, hang over next to a coconut tree or over the pole, so they can collect or harvest the guano. But the way they do is that they put people and also animals into close contact with those bats. And another interface that as an example is a rodent trade, which is popular because people eat rodent, and rodents meat is quite popular and expensive. So people collect rodent from different locations across the country and transport them in a truck or motorbike, pack them together in those different cages that you see here, and then transport them along the road up to 200, 300, 400, 500 kilometers to the border to Vietnam to sell to the other side for consumption. We do also have locations where people consume rats in Cambodia as well. So in this PREDICT program for the last 10 years, since 2009 until 2020, that was implemented in Cambodia, but also in other parts of the world, including Thailand, that you will hear later on from Dr. Supaporn. The main goal of this program is to strengthen global capacity for the detections and discovery of viruses that have the potential to cause pandemic and from animal to humans. What we have learned and what we have gained from this long-term program is that we have our laboratory capacity to the tools at the laboratory to be able to detect not only known viruses but also potentially new viruses that may circulating in different animal species. And importantly also is that we through this program, through this 10 years of work, we have a large biobank of samples that can be used for further characterization, for further studying, and also to look back in those samples in the biobanks to look for any new pathogens that were not detected at the time of the study. And also, as the project was implemented in several countries around the world, we built a large network of laboratories that we can share assays, share process control, share knowledge, share data between each other in order to be ready for any potential threat. On top of that, there is also a training component, One Health capacity building within the countries, including students and also in-service government staff from animal and human sectors. And we also proved that there were an excellent collaborations between humans, animal, and forestry sectors in each country, and it's not well implemented so far, but we have proved that through this program we have helped people from different sectors to work together. And as example from Cambodia here, as a summary is that we have tested almost 80,000 samples from different animal taxa and humans. We have found 35 known viruses, and importantly 40 new viruses from animals and also from humans. We have trained more than about 300 people in One Health workforce. And this is the number of samples that we have collected in our biobank. An example of our field work, collecting samples from different animals, working during the night to collect bat samples. And just an example also what we have detected throughout with the sample we have collected from different animals and humans. In general, we have a wide variety of diversity of viruses from in human here, influenza viruses, avian influenza in poultry, mumps virus, Newcastle virus in ducks. And in terms of coronavirus, as you can see here, there's a wide variety of coronaviruses circulating in humans, in bats and rodents, and also in domestic animals. This is a list of new viruses that we detected mostly in bats, including coronavirus, paramyxovirus, and a number of rhabdoviruses in bats. And this slide just to show an example of virus detections in different specimens. In terms of detections rate in different, we can detect at different rates in different specimens according to the pathogens we tested. For example, we detected a lot of paramyxoviruses in urines or orogenital swab, and this is known that these paramyxoviruses are excreted more in urines. But in general, what we have noticed is that feces or rectal swab present the highest rate of positive rate in virus detection, especially coronavirus here present in orange color. And taking this opportunity as well is that using those biobank samples that we collected for the last 10 years to answer the questions that we are having for the current pandemic of SARS-CoV-2, we use those biobank samples to test, to screen for SARS-CoV-2 that may be circulating in the country long ago. So for that objective, we screened back more than 400 samples collected during between 2010 and 2018 from 6 bat families and 2 carnivorous mammals families. Among those 430 samples, we detected 16 samples positive for coronavirus, 11 belong to alpha and 5 to beta coronavirus. Among the 5, we detected 2 SARS-CoV-2 related to 2 SARS-CoV-2 related viruses in Chamel horseshoe bats in Cambodia, and these 2 samples were collected since 2010 in the province called Siem Trang in the northern part of the country. And comparing the full genome of this virus with the SARS-CoV-2 in humans, the virus detected in Cambodia share more than 92% of identity with the human virus. And this virus, as you see here, cluster in a sublineage close to SARS-CoV-2 circulating in human. And as Dr. Supaporn will present here, the virus that she detected in 2020 in Thailand. The analysis of the full genome of this virus showed that this virus presents numerous recombination events across the genome, and it is not surprising because, as we know, coronavirus circulate or present in bats in high positive rate, and this bat may share this virus in the same colonies, and that's why this virus presents a large amount of recombination in the genome. And our virus cluster closely to as a sister clade to the SARS-CoV-2 and also to the RATG13 and present. If we look at the different location, different region of the genome, our virus sit closely to the SARS-CoV-2 in humans, in this particular region. It seems that the virus from Cambodia take this fragment of genome from SARS-CoV-2, and this is another phylogenetic tree to show the overview of the SARS-CoV-2 detected here in Cambodia in red, and the one that were detected in Thailand recently in 2020. And until present, it seems that rhinolophus bat species are the main host or reservoir of this SARS-CoV-2 ancestor, and different species of this genus harbor viruses that were detected in different countries. For example, here in Yunnan, China, in Cambodia, and also here recently in Thailand. And if you look at the geographic distribution of these bat species, it seems that there is a specific geographical distribution, especially the Shamali, but what we see here, there is an overlapping distributions among the four different species, and the distribution span from southern part of China, including Yunnan and to Southeast Asia, and our study here show that the ancestor of SARS-CoV-2 causing the pandemic, current pandemic, may present largely wider than what we observe recently in China, but also present also in other countries in Southeast Asia. And that another analysis from our colleague from the Museum National d'Histoire Naturelle showed that Rhinolophus shamli, when he compared those found in Cambodia and Laos, he showed that the different species of bat may share the same habitats and also co-roost, and that is why there is a potential of sharing the virus between bat species. And then it is interesting to also to look at the spike protein, because you want to know if the virus detected in bats may be able to infect humans or not. So the analysis from the spike protein, the receptor binding domain from our virus in Cambodia show that they are quite similar. There is some part of high conserved regions in these spike proteins, but show some difference, like here you present here the shortening of the loop at the beginning of the receptor binding motif, and also the fluorescent site that is important for the virus to infect human cell is not present in Cambodian virus, showing that this cousin virus in Cambodia is not able to infect human. And I would like also, besides this, the work that we have done during the PREDICT program, we have also other projects here in the unit working on bats and also rodents. And I want to take the example here, come across a project supported by EU and led by Dr. Julien Capel from CIRAD. It was a project originally designed for Nipah virus work, but taking the opportunity that we have the samples and the pandemic of SARS-CoV-2, we use this sample to study the drivers of coronavirus positivity in bats here in Cambodia. So this specific design of the project is that we have a longitudinal monthly sampling between 2014 and 2016, and the bats were recorded according to the age, the sex, and also reproductive status. In general, we have about, and this study was conducted in two different locations, two different settings. One is a flying fowl roost in Kandahar province, and the other one is the cave, where different bat species were collected from different bat species. But in general, the positive rate of coronavirus in these two locations is about 4 to 4.25% positive rate in the two locations. What is important from this finding is that we observe a significant higher positive rate in non-adult bats, but we do not see any significant effect of the different species of bat or the sex of bat. What is important from that is the seasonality that we observe from our finding. There is a peak of coronavirus circulations about 2 to 6 months after birth period. Means that perhaps in the colony of bats, there are increasing number of juvenile bats being more susceptible to infections, because these juvenile have no more maternity antibodies to protect them, and also they are naive to new infections. So at that time, we see higher circulations of coronavirus in the bat colony. However, due to the limitation of the design and also the sampling timing between years, we see some level of variability between years. So there should be another prospective, more well-designed study longitudinal for 2 or 3 years to understand better and to prove this seasonality in bats. We use the sequence, that coronavirus sequence from these different bats to understand the cluster, and also to see the positive coronavirus positive in different bat species. What we observe is that bat species sharing the same cave for roosting. For example, in Cambodge province, where we have different bat species in the same cave, do not share the same coronavirus. So different bat species carry different coronavirus. And in contrast, for example, here, Infectious lavatus, the same one species, but it carries different coronavirus, different genera of coronavirus. One is alpha and one is beta. And interestingly, coronavirus were detected in bats, in flying fox cluster separately, in one cluster separately. And this is interesting to go further and to try to understand better this coronavirus from flying fox, which is not well known in the literature. So finally, this is my last slide, just a few main messages from this different work for the last 10 years on coronavirus in Cambodia. Is that we have a large diversity of coronavirus circulating in wildlife, and also in domestic animals and human interface. And one have approached surveillance program to monitor potential emergence and spillover is very important to be implemented, and not only in a country specific, but at regional level. What we have learned also is that feces or rectal swab are the best sample for surveillance program if we have no choice or no choice to collect other kind of samples. This is the best sample for surveillance. And one other important conclusion is that the ancestor of SARS-CoV-2 circulating in humans have a much wider geographic distributions outside of China, which is important for the effort of finding the origins of this virus, and Southeast Asia might present a potential hotspot for further investigations. And then from the last work that we have done with Julian is that there is a higher positive rate of coronavirus in juvenile bats between June and November in Cambodia. And then so I would like to stress again that there is an importance of support, funding support for the long-term longitudinal surveillance of not only coronavirus, but also other virus with pandemic potential like Nipah virus, hantavirus in rodents, which is crucial to be prepared ourselves for the next pandemic. And this type of longitudinal surveillance should be one half interface between animal and human surveillance. It's a multiple approaches to monitor any emergence and spillover events from animal to humans, not only in the community, but also in hospital settings. As you know, different bat species carry different viruses. So it's also important not to focus on one bat species, but also to work to collect samples from different bat species that we may find, and also other wild animal it interface with those bats. The way we do, and also the way we do regular sampling, like to understand the seasonality of virus shedding is very important to be able to provide targeted preventions, and also to provide to maximize the probability of detection of the virus, meaning that we just go to one time to the field to collect samples, and we know the time that we have more chance to detect or to detect more viruses from those bats. And with that, I would like to thank for your attention, and also to thank all other collaborators, partners, and also funders that help produce all this data for the scientific community. And thank you.
Thank you so much, Dr. Veasna, for your excellent presentation, and thank you so much for the excellent work you and your colleagues are doing at the Institut Pasteur of Cambodia. This work couldn't be more important to both the current pandemic and also to understanding future pandemics. So thank you so much for this. I will hold off on questions because that will be handled by my colleague, Dr. Supaporn, and I see there are already a number that have come in. Instead, we will move to our next presentation, and then we will pass questions to you, Doctor, after that presentation. So with this, please allow me to introduce our next speaker, Dr. Supaporn, who is a researcher at the Thai Red Cross Emerging Infectious Diseases Health Center, which is a WHO collaborating center for research and training on viral zoonoses. Her research interests include emerging infectious diseases in bats, including Nipah, rabies, coronavirus, and novel pathogens, as well as molecular diagnoses and sequencing of viruses. Her center is responsible for molecular diagnosis services to the hospital and is the reference laboratory for rabies virus, MERS-CoV, COVID-19, Ebola, Zika, and other infectious disease diagnoses for the Ministry of Public Health, Thailand. She has served and consulted on several WHO and Thai government committees. Dr. Supaporn's team was the first to positively identify a human COVID-19 infection outside of China. So Dr. Supaporn is obviously a very esteemed scientist, a researcher, and is doing excellent work, and we're very grateful to have her with us today. So with that, I pass the floor to you, Dr. Supaporn.
Thank you, Alex. I'm sorry that I could not share. Oh, sorry. I could not turn on my video yet, but I can speak, no problem. Yep. Could you see my screen? Okay.
Yes, we can see the screen.
Okay. Okay. This is full screen. I'm not sure. Okay. Yep. First of all, I would like to thank the organizer for inviting me to this very important webinar, and let me allow me to share the zoonotic surveillance work in Thailand. And my topic today is the surveillance of the bats SARS-CoV-2 related coronaviruses in Thailand. The first report of the SARS-CoV-2 related coronaviruses in the Wacharapluesadee has been published by the team from China, Wuhan Institute of Virology, that led by Professor Cheng-Li Shi last year, February last year, and the virus that was found in in the Wacharapluesadee called Rhinolophus affinis that was collected in 2013 in Yunnan. The virus was named RATG13, as Dr. Veasna had mentioned in his presentation. This virus shows 96% nucleotide identity to the human SARS-CoV-2 viruses. And it is interesting that this virus can use the ACE2 human receptor for binding to infect the cell. And the second report, the other report of the SARS-CoV-2 related virus in bat was found in other Wacharapluesadee called the Rhinolophus marianus. And the virus found was named RMYN02. The sample was collected in 2019, and this virus shares 93% nucleotide identity to the human SARS-CoV-2, but it is could not bind to the ACE2, human ACE2. From those two reports, it emphasized that there should be character host and the virus species specificity between the SARS coronavirus and the Rhinolophus. So this lets pop up in my mind that in Thailand there are at least 23 Wacharapluesadee as reported by Thai Department of National Parks, Wildlife and Plant Conservation in 2016. So that means that there should be some SARS-CoV-2 related virus in the Wacharapluesadee in Thailand. So this is why I start the surveillance of the virus in Wacharapluesadee last year. And the surveillance of the SARS-CoV-2 related virus in Thailand has been started in June 2020 after the COVID outbreak in Thailand is better. So then I was allowed to go to the field for bat study. And this study was supported by the grant from BTRP, US DETRA, and from the WHO, and with the strong collaboration with the partner in country, that is Kasetsart University and Department of National Parks, Wildlife and Plant Conservation, and Duong NUS Medical School in the program of emerging infectious disease led by Professor Lin Wang. From this study, 12 trips were conducted during June to December last year. The objective, there are two objectives. The first one, we want to survey for the location of the Wacharapluesadee colonies in Thailand. And the second one is to study on the SARS-CoV-2 related virus in Wacharapluesadee and other bats in these areas. For each trip, we collect 100 bats for each trip, and the blood was taken from the bat wing for the serology study on the human SARS-CoV-2 antibody, and the rectal swab was also collected for the molecular testing and genome characterization. Blood was collected using the capillary tube. 1 to 2 capillary tubes were collected from each bat, and the plasma of bat was separated at the field trip for the serology study. Let me start with the serology study for the SARS-CoV-2 in bat serum, bat plasma. So for the bat serology study, we use the ELISA-based neutralizing antibody assay for detect the neutralizing antibody against SARS-CoV-2 IBD. This technology was invented by Professor Lin Fa Wang lab at Duke NUS, and it is a competitive ELISA between the antibody and the ACE2 receptor that coated at the bottom of the plate. So that means it not need the secondary antibody for as a reporter. So that means it can be used for all animal or human for testing antibody. And this graph shows the percent inhibition of the antibody from one study size that the samples was collected 3 times during the in 2018 in August, September, and November, and 1 time in 2020 December last year. The percent the cut off of the ELISA was 70%, and from this size there are 3 bat species, including the Telephone plicator, Hippocedelus lavatus, Tarphosus melanopicon. You can see that all of these 400 bat plasma samples were negative for antibody against SARS-CoV-2 IBD antigen. And this is the other study on the serology study from the sample from bats that were collected from 4 provinces, 4 sites, including Chanthaburi, Chonburi, Pathum Thani, and Phetchaburi. And this table show the list of the bat species and the number of sites that were collected. And you can see that there are variety of the bat species included in this sub-study. Again, so there is no antibody positive from these 576 bat samples. And this is the other bat colony that the result has been reported last month in Nature Communication. This is bat plasma from the acuminatus horseshoe bat collected at the eastern of Thailand, Chachoengsao province, and from 98, not 100. From 98 bat plasma, 4 samples were positive against the SARS-CoV-2 IBD antigen using the commercial kit called the CPAT kit, the same kit as the previous 2 sites. You can see that 2 of 4 plasma show really high titer, at 87 and 94% inhibition. That means it had very high response against the SARS-CoV-2 IBD. It means there should be some of the SARS-CoV-2 related viruses in this bat colony. So let me move to the molecular surveillance that had been done at the same bat colony, same bat sample. In the rectal swab from the same bat colony. So this is the result. We use the family-wide PCR for the primer for the RDRP gene of the bat to test for the screening of 113 of them were PCR positive. And when we compare the RDRP fragment, gene fragment, we found that it's very close to the human SARS-CoV-2. It's around 95%. But when the whole genome was conducted from one batch, from one samples, the percent identity was dropped to 91%. And you can see that here at the graph here, most of the genome is similar except at the spike gene. And this is, you can see from this table also that the virus called RACCS203, let me call 03, there's just only 71% identity in the spike gene when compared to the human SARS-CoV-2 or the RATG13 in China. But it has very high similarity to the RMYN02 from China also. When we compare the RBD nucleotide sequence with the other bad SARS-CoV-2 or SARS-related coronaviruses, it's found that our the 203 virus belong to the non-S2 usage. Why the RATG13 belong to the S2 usage with the grouping with the pangolin and the human SARS-CoV-2. And it's confirmed on your right hand graph that this is the binding detection interaction between the pseudotype of the virus and the S2. It's found that it's confirmed that the CS203 and the YN02 could not bind to the human S2. And this table summarized the bad coronavirus and the S2 usage. As I mentioned earlier, that RATG13 from China use the S2 to bind to the human cell, and the other virus is called RC319 from Japan. Even though the percent identity is just only 89%, but it can use the S2 to bind the human cell. So this graph is to summarize where the bad SARS-CoV-2 related virus had been found. It had been found in China, in Hubei, Yunnan, and it had been found in Japan, and Thailand, and Cambodia. And this is the summary of the PCR results that were conducted from other bat species. This from 13 bat species collected in 4 provinces in Thailand during year 2013. During last year 2020. Only you can see that this is the coronavirus species with in different color, and this is the bat species and the number of the tests. You can see that for the Rhinolophus acuminatus, it found just only the Sabico virus, as I already present to you earlier. And for other bat species, you can see some of them are really interesting. There are more than one coronaviruses found in one bat species. For example, the Hypocidalus lavatus and the Cherryphon plicata, for example. So it is very interesting that there are several, I can say many of the coronaviruses found in bat in Southeast Asia. So for the conclusion, the whole genome of the SARS-CoV-2 related virus was successful obtained using the next generation sequencing, and the high identity of the Thai virus, I should say 203 virus to the virus found from Malayan horseshoe bat in Yunnan in China can extend the knowledge of the origin of SARS-CoV-2 in bat. And our study extends the geographic distribution of the genetically diverse of the SARS-CoV-2 related coronaviruses from Japan, China to Thailand over the 4,800 kilometer range. And the SARS-CoV-2 neutralizing antibodies were detected in the acuminatus horseshoe bat, but not other bat species in this study. It can say that the serology surveillance can be also the critical for tracing the animal origin and the animal to human spillover events. And the more SARS-CoV-2 related coronaviruses are likely to be discovered in the region if we can have conduct the surveillance in our countries, in our regions. And Southeast Asia is a large and diverse bat population that may be more likely to a hotspot for such viruses. My last but not least slide is to thank the team, the few team that led by Dr. Patip Duong and Dr. Wacharapluesadee Manion. Dr. Patip he's working at the Kasetsart University at the Department of Forestry, and Dr. Wacharapluesadee he's working at the Wildlife Veterinary at the Department of Wildlife, Thailand, and the laboratory team led by Professor Thiravat Hemajuthar and the staff who are working together to carry the multidisciplinary from the field to the lab and from the lab to the field. And I would like to thank our sponsor at the beginning of the bat research in Thailand, USAID, EcoHealth Alliance, and thank BTRP, US DETRA to support the field trip during last year, and the partner, Department of Wildlife, the Kasetsart University, and WHO, and Duke NUS Medical School to for the technical support to let all the work more deep and can clarify on some research question that bring our research study to the global level. Thank you for your attention.